Let's be honest. When you hear the word cloning, your mind probably jumps straight to some sci-fi movie, right? An army of identical soldiers, maybe a perfect copy of yourself walking around. But the reality of animal cloning today is, well, way more fascinating and honestly, a lot more practical than you might think. It's a story that starts not in some futuristic lab, but down on the farm, and it's revealing some absolutely incredible secrets about what our DNA really controls. All right, let's get into it.
So, first things first, why would anyone even want to clone an animal? It's not really about creating an identical pet, at least not for the industry that's pushing this technology forward. The real motivation is actually tied to something humans have been doing for centuries, agriculture, and that endless quest for the perfect animal.
You know, the US Food and Drug Administration actually has a great way of putting this. Just imagine you're a farmer and you have this one unbelievable cow. I mean, she's a total superstar, healthy, fertile, and just an amazing milk producer. She basically won the genetic lottery. But she's getting older, and that amazing combination of genes, well, it could be lost forever. Her babies are always a genetic mix, never a guarantee.
And that, right there, is the million-dollar question. What if you didn't have to roll the genetic dice every single time? What if you could just take the exact blueprint of your very best animal and, well, just make another one? This is the exact problem that agricultural cloning promises to solve.
Okay, so this isn't magic, and clones aren't just grown in a test tube from scratch. It's a really precise biological process. We're going to break down the main technique used today, and you'll see it's basically a super advanced version of reproductive tech that's been around for decades.
The gold standard for cloning is a method with a mouthful of a name, somatic cell nuclear transfer, or SCNT. But let's break that down. A somatic cell is just any old body cell, like a skin cell, for example. It's not a sperm or an egg. And nuclear transfer just means we're moving the nucleus, the part of the cell that holds all the DNA. So basically, we're transferring the DNA from a body cell into an egg cell. Simple as that.
So here's how it goes down. First, scientists take a donor egg and scoop out its nucleus, leaving this empty little shell that's ready for new instructions. Next, they take a body cell from our prize cow and pull out its nucleus, which has her complete DNA blueprint. Then, they pop that nucleus into the empty egg. A little zap of electricity helps it all fuse together and tricks the egg into thinking it's been fertilized, so it starts dividing. This brand new embryo is then placed into a surrogate mother, and voila! The result is a genetic twin of the original cow, just born at a later time.
And this really shows you the key difference, right? With natural reproduction, you get a genetic shuffle from two parents. The outcome is always going to be a little bit of a surprise. But with SCNT cloning, you've only got one genetic parent. The offspring is a direct genetic copy, which gives breeders this incredible, unprecedented control over the traits they want to keep.
Now, with this powerful tech, you might be imagining farms just full of identical cloned animals, but that's not really how it's used. See, cloning is about building a better herd, not just a copied one.
So this is a super important point. Farmers aren't cloning animals to turn them into steaks and milk. Clones are way too valuable for that. Instead, they become these elite breeding animals. You clone your prize cow to get a few more prize cows, and then you use them for good old-fashioned conventional breeding. It's their naturally conceived offspring that enter the food supply, which rapidly improves the entire herd's genetics.
And what are they breeding for? Well, all the stuff that makes farming more efficient and sustainable. I mean, imagine creating a whole herd that's naturally resistant to common diseases. Think of the money saved on vet bills. Or animals that are perfectly happy in hotter, drier climates. Or that consistently produce higher quality, leaner meat that people actually want to buy. Cloning is basically a shortcut to lock in these amazing genetics and spread them way faster than you ever could before.
Of course, anytime you talk about new food technology, the first question on everybody's mind is a simple one. Is it safe? The idea of eating meat or drinking milk that's connected to cloning, yeah, it understandably raises a few eyebrows.
And the US Food and Drug Administration took this question very, very seriously. Way back in 2001, as this tech was getting ready for prime time, they asked the industry to voluntarily keep food from clones off the market and launched a massive, multi-year risk assessment.
And when I say they looked into it, I mean they really looked into it. This wasn't just a quick look. We're talking about analyzing hundreds of scientific articles, getting their hands on the actual health records and blood work from pretty much every single cattle clone in the United States, and comparing them directly to their non-cloned herd mates. They even looked at data from all over the world.
So after years of this exhaustive review, their conclusion was crystal clear.
The FDA stated that food from clones of cattle, pigs, and goats is just as safe as the food we're already eating. They found absolutely no real difference in the health of the animals or the makeup of their meat and milk. The science was settled.
Okay, so just when you think you've got the story of cloning figured out, agriculture, safety, check, check, it takes this absolutely fascinating turn. Some of the most unexpected insights are now coming from a completely different animal, one we have a very unique relationship with, the dog.
Believe it or not, over 1,500 dogs have now been cloned, mostly at one research center. And this massive, unprecedented project has created this incredible natural experiment. With so many genetically identical animals running around, researchers started noticing, well, things that just weren't supposed to happen if clones were truly perfect carbon copies.
The first major shocker was sex reversal. Get this, they had clones that were genetically male. They had the SRY gene, the main boy gene on the Y chromosome, but they were born with female anatomy. The genetic blueprint screamed male, but the physical animal was female. How does that even happen?
And it didn't stop there. Then they saw cleft palates. But the really weird part was that in a litter of genetically identical puppies, one might be born with a cleft palate while its identical twin clone was perfectly fine. If it were purely genetic, they should all be the same.
They also saw these cases of muscular hypermyotrophy, which is this extreme, abnormal overdevelopment of muscle tissue. And again, this would just pop up in some clones, but not others from the very same donor. This condition was often fatal, which shows you that these variations could have really serious health consequences. Clearly, something beyond the raw DNA code was pulling the strings.
So what on earth is going on here? If you have genetically identical animals developing differently, it forces us to look beyond the sequence of A's, T's, C's, and G's. It turns out our genes are not a simple instruction manual, they're more like a complex musical score, and something else is acting as the conductor.
And that conductor has a name: epigenetics. This is the crucial piece of the puzzle that explains why these cloned dogs, even with identical DNA, could turn out so physically different from one another. So epigenetics is basically a layer of chemical marks, or tags, that sit on top of our DNA. Think of them like little sticky notes or light switches. They don't change the genes themselves, but they tell the genes whether to turn on or turn off in a particular cell. During natural development, these patterns are set very carefully. But that SCNT cloning process, ripping a nucleus out of an adult cell and shoving it into an egg, it's a bit of a shock to the system. The reprogramming of all those little epigenetic switches isn't always perfect. Some switches that should be off get stuck on, and vice versa.
So the bottom line from all of this is that our genes just are not the whole story. A clone is a genetic copy, yes, but it will never be a perfect physical or behavioral copy. These epigenetic patterns, the environment in the womb, nutrition, and just pure random chance during development all play a huge role. And in the funny way, the mistakes in cloning are actually a gift to science. They're giving us this powerful window into the super complex world of epigenetics, helping us understand how our genes are really controlled. And that leaves us with a pretty deep final thought. We often talk about being programmed by our DNA. But if even perfect genetic copies, made in a lab, can turn out so differently, it really challenges that whole idea of genetic destiny. It suggests that who we are, our traits, our health, our very identity, is a much more dynamic and complicated story than what's just written in our genetic code.